This conference will now be recorded.
Okay, so we'll see the video first, then we'll move on to the slides. Okay, so I hope the screen is visible.
Where does bread get its fluffiness? Swiss cheese, its holes. And what makes vinegar so sour? These foods may taste completely different, but all of these phenomena come from tiny organisms chowing down on sugar and belching up some culinary byproducts. Let's start with yeast. Yeast are single-celled fungi used to make bread, beer, and wine, among other products. Yeast break down carbohydrates like sugar to get energy and the molecules they need to function. They have two different ways to do this, the oxygen-dependent or aerobic pathway and the oxygen-independent anaerobic pathway, which is also called fermentation. When you bake bread, yeast can use both pathways, but they normally prefer to start with the anaerobic process of fermentation. In this process, ethanol is produced in addition to CO2. No bread is an alcoholic. Small amounts of alcohol that are secreted evaporate during baking. In the aerobic, oxygen-dependent pathway, the yeast consumes some of the sugar and produce carbon dioxide gas, or CO2, and water. In both processes, the CO2 accumulates and creates tiny bubbles. These bubbles get trapped by gluten and create a sponge-like structure that gives the bread its soft texture. Wine also relies on yeast, but a winemaking setup keeps the oxygen levels low so that yeast consumes sugar using fermentation, the anaerobic pathway. The process often starts with wild yeasts already hanging out on the grapes. But to get consistent results, most winemakers also add carefully selected strains of yeast that can tolerate high levels of alcohol. The yeast consume the sugar in the grape juice, and as the sugar level drops, the alcohol level rises. This doesn't necessarily mean that sweeter wines have less alcohol. Different types of grapes start with different amounts of sugar, and sugar can also be added. What happens to carbon dioxide? It just bubbles away and prevents. In carbonated alcoholic beverages like champagne and beer, sealed containers are used in primary or secondary fermentation to keep the carbon dioxide in the bottle. Wine also introduces us to our second type of food-producing microorganism, bacteria. A special strain of bacteria turns a tart compound in grape juice into softer tasting ones that are responsible for some of the flavors in red wines and Chardonnays. Another type of bacteria, called acetic acid bacteria, isn't so desirable in wine, but they have their function too. If there's oxygen around, these bacteria convert the ethanol in wine into, well, acetic acid. Let this process continue, and you'll eventually get vinegar. Bacteria are the key for cheese, too. To make cheese, milk is inoculated with bacteria. The bacteria gobble up the lactose, a kind of sugar, and produce lactic acid, along with many other chemicals. As the milk gets more and more acidic, its proteins start to aggregate and curdle. That's why spoiled milk is clumpy. Cheesemakers usually add an enzyme called rennet, naturally found inside of cows, goats, and some other mammals to help this process along. Eventually, those little kernels turn into bigger curds, which are pressed to squeeze out the water and create a firm cheese. Different strains of bacteria make different kinds of cheese. For example, a species of bacteria that emits carbon dioxide is what gives Swiss cheese its characteristic holes. Some cheeses, Brie and Camembert, use another kind of microorganism too, mold. So your kitchen functions as a sort of biotechnology lab made by microorganisms that culture your cuisine. Yogurt, soy sauce, sour cream, sauerkraut, kefir, kimchi, kombucha, cheddar, challah, pita, and naan. But maybe not all at the same dinner.
Okay, so we'll go to the slides now. I hope I'm audible to everybody. So we'll discuss a little bit about food bacteriology today, and we'll include the bacteria that can cause harm also. So microorganisms are important in many different ways. They can be pathogenic or disease causing, and these can cause illness once you consume a food. Then they can also cause spoilage, that is, the food may smell, taste, or look unacceptable. And some organisms, as we saw in the video, they can produce a desired food product, that is due to fermentation. So there are different types of organisms which can grow at different temperatures. These we have discussed in general bacteriology before, but just to recap, we'll just go over them once more. Some are saprophiles, that is, they grow at a very low temperature. Phile means loving, and sapro is lower temperature. So naturally, they grow at a very low temperature, like Pseudomonas, Flavibacterium, alkaligens. And why these are important is because these organisms, even though we refrigerate certain food materials, they can even grow on them. Some other organisms are psychotrophic, that is, they can grow best at 37, but can also grow at refrigerated temperatures, like Listeria monocytogenes, which can be of harm once you consume it in a food. Then there are mesophiles, that is, the optimum temperature is 20 to 40 degrees Celsius. They contain most of the human pathogens, like we have been discussing since we have been discussing about systematic bacteriology, like Escherichia coli, Salmonella, Clostridium, etc. Then there are thermophiles, that is, temperatures at more than 45 degrees Celsius, like Bacillus stereothermophilus. Excuse me. So lower temperatures, that is, even in refrigerated temperatures, usually the organisms are either their growth is stopped or their growth is slowed down. So freezing usually helps to stop bacterial growth, unless that organism is like Listeria. So it can also grow, as you might remember that we actually grow them once we try to enrich them in cold enrichment, that is, we grow it at 4 degrees Celsius, in which it can actually grow. Then there are higher temperatures, like in heating, which usually destroys organisms for immediate service foods. That is, everybody, your mother, your grandmother, everybody must be telling you that you have to have food which is hot. So thermal processing of shelf-stable foods destroys the bacteria for longer shelf-life foods. That is temperature dependent upon product acidity. So usually canned foods that are available, so they are inactivated. Clostridium botulinum and others are inactivated, and then you have to heat them up. Okay. So what the disadvantage is that once you heat them up, once the spores are there, those spores might be activated. So that is why usually you should not cool down food. You should not keep food for a longer time and then heat it up and then eat it. Okay. So usually you might be remembering like bacteria divide by binary fission. So just to remind you of that. So this is how they divide. Usually every 20 to 30 minutes they divide by binary fission. Once a wall is formed upon in between the daughter cells, that is the mesosomes, that are the folded type of cell organelles which are present in bacteria, which are actually a folding of the cell membrane, plasma membrane. So in which what will happen is a wall sort of thing will form, and then they will divide. So this is how they divide. They divide by binary fission. So what are important for division of these microorganisms is the availability of their food, the type of acidity or the pH of the food, then the temperature, the time, oxygen, and the moisture which is available. So different organisms need different types of these things in order to grow. So food that is nutrient contents, usually they require, naturally they require water, they require a source of energy, a source of nitrogen, they require vitamins and related growth factors and minerals like we do. So naturally the optimum pH for growth is usually 6 to 8, and the disease causing bacteria usually grow at a range of 4.6 to 9.5, and spoilage bacteria usually they can grow at a very higher range of pH. So this is the pH range you can see. So molds, yeasts, lactic acid bacteria, Staphylococcus aureus, Salmonella, Escherichia coli, Campylobacter, and Vibrio species. So here you can see there are different acidity of foods, that is higher acidity foods and lower acidity foods, which can be inhibited by acid, and that is why we need heat and different type pH to destroy these organisms. So temperature, like optimal growth, like just now we discussed, thermophiles, which like hot conditions, mesophiles, which like warm conditions around the body temperature, but psychrotrophs, psychrophiles, they can grow at refrigerated temperatures. So based on these things just now we discussed, so psychro, meso, thermo, and trophic is they grow or they love to grow during that time, and duric is they can withstand, philic is preferred or they love that temperature. So here you can see these are psychrophiles. So psychrophiles, like we discussed in the previous general bacteriology chapters, you might remember some of the psychrophiles, they can grow at very low temperatures also, like underneath the sea. So at very low temperatures also some of the psychrophiles can grow, but because we are talking of food bacteriology, we are talking about the temperatures in which even if you refrigerate, even canned foods which follow a cold chain, even then these organisms may persist and they may carry onto from the farm to your fork. Okay? So these are the psychrophiles, then mesophiles, thermophiles, and hyperthermophiles. Usually in food, hyperthermophiles are not that much important, but even if some dried or other preserved items are available, even in very high temperatures, the spores may persist, or these hypothermophiles may also persist. So this is the common cooking temperature, the warming temperature, and these we can see in which rapid bacterial growth or dish sanitization, lowest temperature which is required. So there is a huge range in which even organisms can persist even if you just warm up the dish. So here you can see this is in this temperature usually organisms can grow. So that is why we try to either preserve food at very low temperatures or very high temperatures. So how to control growth? Usually temperatures lower than 41 degree Fahrenheit, that is bacteria cease to multiply, but they do not die. So because freezing can cause cell wall damage. As you remember, they have a cell wall of peptidoglycan. So if you freeze them too much, they can cause damage to the cell wall. So that is why they usually keep food preserved by freezing them. Then temperatures higher than 135 degree Fahrenheit, that is if you heat them for a sufficient time, like you might remember about pasteurization, you might remember about very high temperatures in which some of the items are kept and then they are prepared and then they are brought to you. So increased destruction is there with longer times and higher temperatures. So time is under optimal conditions. Some bacteria can double every 20 minutes, like we discussed just now by binary fission. Many organisms, they can divide every 20 minutes. So the colder the storage temperature, naturally the potential. So potentially hazardous foods should not remain in the danger zone for more than 4 hours during the food handling process. So that is why whenever food handling industries and other industries which carry on this thing, so usually they maintain this longer time periods between the danger zone in which the bacteria can multiply. They do not maintain it at that particular temperature. So you can see that generation time, that is the time taken for one bacterium to become two, it is varied in different organisms. So it is in Bacillus cereus. Why Bacillus cereus? Have you? I hope you remember that in fried rices and all Chinese items. So Bacillus cereus is a major important cause of food poisoning. So it requires 28 minutes for generation time. It is one bacterium to become two. Then Escherichia coli about 12.5 minutes. Staphylococcus aureus, so it can also cause toxic shock syndrome. It can also have enterotoxins, and they can have different preformed toxins that can be there in Staphylococcus aureus, like we discussed in our diarrhea chapter. So this Staphylococcus aureus can form a preformed toxin, and it can cause food poisoning within 1 to 6 hours. So Mycobacterium tuberculosis is also an important cause of transmission through food. That is, suppose you are taking raw milk or milk that is not properly pasteurized, or if the source is contaminated, or suppose the person who is bringing it to you is having some disease. So you can have contamination of the milk through many different sources. So as you might remember, we had discussed about bacterial growth phases, the bacterial growth curve. You might remember that there was a log phase, then there was a lag phase, a log phase, then there was a stationary phase, then there was the phase of decline. So we have to remember that these are the different phases in which the bacteria multiply. So that is why it is important that we maintain the bacteria in such a temperature, such a pH, and in such a environment so that the bacteria do not multiply. So based on oxygen requirements of bacteria can be aerobic or anaerobic, like we discussed in the video. So there can be aerobic cycles or anaerobic cycles also for the same organisms. Even if they are facultative organisms, they can grow with or without oxygen. May not be that much abundantly, but even then they can. So here you can see oxygen anaerobic. If you keep them in a test tube in which you have filled it with broth, so what happens is the anaerobic organisms tend to grow at the bottom of the tube, whereas aerobic organisms, wherever oxygen availability is maximum, they grow at the surface, whereas facultative anaerobes can grow throughout the test tube. So this is just, these all slides are just a reminder of what we have already discussed in your previous systematic and general bacteriology. So water activity, that is moisture, that is the measure of free water which is available to the microorganism for growth. As we contain almost 80% water, even the bacteria also contain a lot of water. So that is why this is also affected, the activity of water is also affected with the presence of solutes, that is sugars or salts. So lowering this will reduce the availability for microorganisms to grow. As you remember, your mother or grandmother might be preparing pickles. So you put in a lot of salt, you put in a lot of oil, you put in all those things. Why? So that you put it in the sun for a long time. So your mother might be preparing the mangoes in that way, putting a lot of salt and then putting it in the sun. So that what will happen is these organisms will lose their water content and then they can't persist. So that is why mango pickles will stay for almost a year or more. So that is why, because the organisms cannot survive under a lot of salt or sugar or even reduced water content. So moisture in the food system is always available for the microbial growth and chemical reactions. That is the relative humidity of a food. So this is the moisture required for most foods. So when the water activity is reduced, naturally the organisms cannot grow that. So here you can see that different bacteria, so different foods, they are preserved under that much water content. So meat, fish, sausage, milk, they contain how much water availability, accordingly how much organisms can grow. So bacteria, yeasts, molds, and once the water availability is reduced, so water activity is reduced, what will happen is no microorganisms can proliferate. For example, dried cookies or crackers or bread crusts in which the water content is very low. So you can remember that fresh fruits, meats, fish, vegetables, cheese, spreads. So they have that much water availability in which most bacteria can grow. Whereas dried milk, dried vegetables, cereals, crackers, so Kellogg's, cornflakes, and all which is available to you. So usually they tell you that keep it in a cool and dry place. So usually you keep it in a cool and dry place. So how to control growth? So crackers or biscuits, cookies, so they have a reduced water availability. So that is why whatever foods are available like fresh fruits or meats and all those things, they have lots of water content. So that is why you have to consume them as soon as possible, or you have to preserve them in some way. Otherwise, jams, jellies, or pickles, or the other dried food items, they can stay for a long time. So microorganisms can grow on food and in the environment under right conditions. And we remember about the different factors which are important for the growth of microorganisms, like food, the availability of food, the acidity, the temperature, time, oxygen, and moisture. So fat, Tom. So fat, Tom, you can remember these are the different factors in which the microorganisms can grow. Okay. So how to intervene? So naturally, how to intervene? There are different interactive effects of factors which affect microbial growth. that is additive effects, synergistic effects, and antagonistic effects. So what do? We have usually in the food industries, they have ingredients that have very good specifications and control of the incoming ingredients. That is, they have an adequate thermal process to destroy the microorganisms. They have an adequate packaging system to protect the product, and after processing, they control the distribution and the system for tracking and recalling. That is, usually once you buy something, they will have written that this is the time in which this food is good. Otherwise, if it is expired, you should not consume it. So suppose some item is the package is off. So those items you should not consume. So there are different food safety and quality programs like the HACCP. So we have discussed about the HACCP in a different class also. So sanitation and food quality programs are there which maintain this. So there are certificates, there are letters of guarantee which guarantee that the food is good. So thermal processing like cooking, pasteurization, and commercial sterility, that is aseptic processing and hot filling. So package integrity, that is, suppose you have brought a canned item or a jam bottle, so they have a hermetic seal. So they have a vacuum seal in which visual inspections and top measurements are there, in which you can ensure that your food is good. Okay. So there are low acidity foods like which have a process to eliminate different spores of Clostridium botulinum. For example, most meat, vegetable and dairy products and acidified foods which are not required to have a process to eliminate the Clostridium botulinum, like pickle products or mayonnaise, and they often are processed with pasteurization or hot filling, and high acid foods which are not required to have a heat process like sliced oranges or condiments, and they are also preserved under pasteurization or hot filling. So different pathogens are there which naturally organisms are ubiquitous. They can be harmless or even beneficial, but they can cause spoilage. That is, the food becomes inedible due to changes in color, flavor, odor, or appearance or texture, and they grow to high levels and break down the food components. So that is why what happens is you have to eliminate the pathogens which can cause foodborne illness, and which there are certain indicator organisms like you might have discussed already, the coliform counts, that is the Escherichia coli. Suppose you are taking a glass of water, so that water has to be standardized, that is, it has to be of the drinking standard. So Escherichia coli counts have to be not there, so that you can drink that water. So coliforms do not cause illness, but they are indicators that fecal contamination has taken place. So the foodborne hazards are there which can be made unsafe for human consumption. So that is why food quality controls are there. So different food poisoning or foodborne illnesses. There are many illnesses per year, about 46 million illnesses a year at least, and many hospitalizations, deaths, and a lot of money which is lost in pain, suffering, reduced productivity, and medication costs. So there are common foodborne pathogens like bacteria. You might remember there are Escherichia coli, the hemorrhagic Escherichia coli, that is O157H7, then Salmonella species. Many Salmonella, apart from typhoid, they can cause gastroenteritis. There can be Staphylococcus aureus, which produces an enterotoxin. Just now we discussed. There is Listeria monocytogenes, which can be transmitted through food, even refrigerated food. There is Campylobacter jejuni. There is Shigella species, which can be transmitted through food. Then there are viruses like noroviruses, rotaviruses, and hepatitis A viruses, which can be transmitted through food. So fecal-oral transmission is there. So many of these viruses can cause diarrhea, and hepatitis A can cause hepatitis. So parasites like Cryptosporidium parvum, Giardia lamblia, Cyclospora, etc. So all of them can have fecal-oral transmission. So about 80 to 90% foodborne illnesses come from at least these 4 bacteria, that is Campylobacter, Salmonella, Clostridium perfringens, and Staphylococcus aureus. So infection, that is microorganisms are ingested and then cause illness, that is toxins are produced by the pathogen, and then once the food is consumed, illness can occur. So even if the organisms are killed, the toxin can still remain in the food. So even if you eat it, the toxin may also persist still. So this is enterohemorrhagic, so Escherichia coli, then there is Salmonella, there is Listeria, Campylobacter, Staphylococcus aureus, and Clostridium botulinum also, which can cause food poisoning. So here you can see that different cases and different deaths can be there due to these organisms. Hemorrhagic colitis can be there due to Escherichia coli O157H7. So hams and other food preserved items, canned foods. So just you heat them up and then you take it. So those items, if the cold chain little bit, if there is any lapse, so these organisms can grow and then they can cause diarrhea, which can be bloody diarrhea. They can cause HUS, and then the contaminants can be like milk, meat, fruits, vegetables, and water. So salmonellosis, that is, many of them can cause foodborne illnesses. The incubation period is a little bit longer, and the nausea, fever, diarrhea, arthritis, so all these can be there. So Listeria monocytogenes can cause listeriosis and can be transmitted through meat or milk, and then it can cause meningitis, septicemia, and even miscarriage because it is transmitted from the mother to the baby. So Campylobacter jejuni can cause campylobacteriosis, it can cause nausea, fever, and diarrhea, and it can be transmitted through milk, meat, and water. So Staphylococcus aureus, as you might remember, it causes an enterotoxin, it causes intoxication. The incubation period is very short because the preformed toxin can be there in the food. It can cause nausea, fever, and diarrhea, and milk, meat, and eggs can be the positive food items. So botulinum toxin, it can cause botulism and can cause intoxication, and it can cause difficulty in speaking, swallowing, or breathing. And contaminants can be foods which have a higher pH or low oxygen foods. And you might remember about the Rose Bengal agglutination test, which is used for testing sera or even milk. So here we have the Rose Bengal test. It is an agglutination test in which the rapid diagnosis can be done if there is contamination with Brucella. So Brucella abortus smooth cells are there, a suspension is there, which is stained with a Rose Bengal pink colored dye, which is used to detect Brucella agglutinins. So if the antibodies are present in the milk or in the serum of the patient, so once you mix it, so if there is Brucella antibodies, so there will be agglutination. So just to remind you, these classes are all like a revision of what you have previously discussed. So this we have discussed previously in brucellosis. So including pasteurization, you might remember there is the heating and then the cooling. So you heat it to a temperature of 72 degrees Celsius, and then you rapidly cool it to 15 degrees Celsius. So that is how you do pasteurization. So that is how milk packets are available to you after they are pasteurized. So how to prevent foodborne illnesses? First is you have to cook. So all the meat, poultry, so do not take partially cooked or raw or smoked fishes, smoked items you should not take. After they are properly cooked only you have to take. So meat, poultry, eggs, so you have to cook it to at least 160 degrees Fahrenheit. So other than spore forming bacteria, all the other bacteria, viruses, and parasites are killed quite easily if you heat it to 160 degrees Fahrenheit. So how to avoid cross contamination? So do not cross contaminate one food with another. The raw food is kept separate from the cooked product. Then you have to chill food. So if you have a refrigerator, you should not refrigerate for a very long time, but till you eat, the food has to be kept chilled, especially in the weather that we have here in tropical climates. So if you have cooked it, so most of the organisms might have died, but spore formers can be still they can be alive. So that is why they are resistant to cooking. So they can be very active and can proliferate rapidly at the temperature that we have here. So room temperatures it can proliferate. So that is why you have to keep foods chilled. And then you have to clean food, especially after the COVID situation. You might remember your mother might be washing the vegetables or food items with salt water or with vinegar water, and you have to maintain personal hygiene. So you have to wash your hands regularly, wear hair nets, plastic gloves, etc., so that the spoilage organisms like Erwinia, Pseudomonas, Flavobacteria, lactic acid bacteria, they or fungi, they do not grow in your foods. So thank you all for patient listening. So we'll see a short video after which we can discuss today.
Food technologists cannot be a food technologist if they do not know food microbiology. In this series of videos, we will understand in detail about pathogenic microorganisms. What are the primary sources of that particular pathogen? What are the root sources? Pathogenicity, contributing factors, atmosphere, sport or non-sport form, temperature, pH, and water activity required for the growth of that pathogen. And in this part, we are going to cover the details about first pathogen, that is salmonella. Before we jump into the details of salmonella, just quickly to know the basics of microbiology. What are microorganisms? Technically, a microorganism is an organism that is microscopic. Microorganisms can be bacteria, mold, yeast, or fungi. There are two categories of microorganisms: spoiling microorganisms, microorganisms. Spoiling microorganisms are either bacteria or fungi, and these are too small to be seen, that cause food to deteriorate and develop unpleasant odors, tastes, and textures. Whereas pathogenic microorganisms include dangerous cases of food poisoning and pose a serious health risk to consumer. In short, organisms which do not cause diseases and just spoil are called non-pathogenic. However, pathogens can make us and may cause death. After this understanding, presenting here the list of pathogens. This is the list of main pathogens: salmonella, Listeria, Clostridium, Staphylococcus, Lactobacter, Legionella, E. coli, Shigella, Bacillus cereus. Salmonella. Salmonella is ranked among the most important initiators of cases of food poisoning. The WHO estimates more than 16 million global infections per year, and more than half a million cases are fatal. Primary source. Primary source of salmonella is intestinal tract of people and animals. Source. Salmonella are found in raw food such as eggs, meat, and milk, and should only be slightly heated or not at all heated. Example: raw milk cheese, raw sausage, chocolate, ice cream. It can also be present in nuts, spices, flour, etc. Pathogens. An infection from salmonella can cause nausea, vomiting, diarrhea, fever, and headache. Contributing factors. Cross-contamination, underfood, or agriculture practice, environmental contamination are the contributing factors. Atmosphere. Atmosphere required for salmonella is facultative. That means salmonella can grow and stay in both aerobic and anaerobic environment. Spore/non-spore forming. Salmonella is non-spore forming microorganism. Temperature. Temperature for salmonella to grow is 6 to 47 degrees Celsius, when the optimum temperature is 35 to 45 degrees Celsius. pH. Salmonella can grow having pH between 3.7 to 9.5. Water activity. Salmonella requires greater than 0.94 water activity to grow. This is all brief about salmonella. We'll bring next video on another pathogen and apply the same basic aesthetics.
So thank you for joining us today. I hope you and your families are all safe. Take care. Thank you.